Good afternoon. On behalf of everyone here at the Center for Brain Health, welcome to the Frontiers of Brain Health, where we take a deep dive into the most exciting new developments in brain research. I'm Dr. Jane Wigginton, the Medical Science Research Director here at the Center for Brain Health, and I'm excited to be your host for this fabulous presentation. I want to let you know up front that we'll have a few minutes at the end to talk about some questions. So if you're visiting with us virtually, please use the Q&A function. And if you're here in person, please have your questions ready to ask at the end. We'll get to as many as we can. Also, this presentation is being recorded. And if there's anyone here who doesn't know much about us, I want to let you know that the Center for Brain Health is a cognitive neuroscience research center of the University of Texas at Dallas. And importantly, for the past 3 decades, we've been leading the scientific quest to measure and improve brain health across the lifespan. Because here's the bottom line. Optimal brain performance is the newest and most important frontier of human potential. So now, without further ado, it's my distinct pleasure to introduce you to our speakers. We actually have 2 today who are wonderful colleagues and collaborators on an innovative and exciting new project that we're embarking on. And first, I'd like to introduce Dr. Cristin Welle, who's a systems neurophysiologist with expertise in the interaction between medical devices and the nervous system. She serves as associate professor in the Department of Physiology and Biophysics, as well as vice chair of research in the Department of Neurosurgery at the University of Colorado Anschutz Medical Campus. Her research explores how vagus nerve stimulation, or VNS as we know it, paired with motor learning enhances skill acquisition by modulating phasic cholinergic signaling and cortical plasticity. Interestingly, she's exploring mechanistic evidence that VNS can enhance oligodendrogenesis and restore myelin deficits to improve long-term motor function. And next, I'd also like to introduce you to Dr. William, or Jamie as we know him, Tyler, who's a professor of biomedical engineering in the School of Engineering, as well as senior scientist in the Comprehensive Neuroscience Center and professor in both the Center for Clinical and Translational Science and Physical Medicine and Rehabilitation in the School of Medicine at the University of Alabama at Birmingham. He's an academic entrepreneur and subject matter expert in neuromodulation and human performance enhancement. Don't we all want some of that? He's significant experience in leading academic, industry, and government research teams in medical device design, engineering, and translation, as well as other methods of psychophysiological and neurophysiological regulation, and he has expertise, very interestingly, in psychotropic and psychedelic drug development. And now, without further ado, I'm going to turn it over to Dr. Welle. Sorry, to Dr.
I'm going to take her place. Thank you, Jane. Sorry, I don't want to burn your retina. Thank you, Dr. Sandy Chapman, for inviting me here to give this talk. I'm very honored to be here. I've heard a lot about the Center for Brain Health for many years. And you have lovely facilities and a wonderful team, so thank you for inviting me. I'm going to start the talks by talking about vagus nerve stimulation for enhancing plasticity and brain health. In the interest of disclosing my financial interest, I am a co-founder of a company, IST. I'm also a co-founder of Diamond Therapeutics. I have issued and pending patents on medical devices for neuromodulation and 5-HT agonists. Today I'll discuss modulation of autonomic nervous system activity by transcutaneous electrical stimulation of the external ear. So I'm going to tell you, I guess, a story. It's more of an entrepreneurial journey than a scientific story. This is the picture in 2012. This was kind of our iconic roadmap, if you will. We had just developed a trigeminal nerve stimulation device. It's trigeminal vagal, and hopefully that's one of the lessons you learned today is that trigeminal and vagus nerve have a lot in common. They have some distinct features as well, but we developed this trigeminal nerve stimulation device and wanted to focus on brain health and said, well, what are the things that we could do that would affect a brain and make it more healthy? So reducing stress, improving sleep quality, increasing energy, encouraging socialization, enhancing focus, boosting motivation. I think you all know this. The experiments that we ran at the time, you'll never find in academic journals, but they were very revealing to us, and we learned a lot. So this is an example of different experiments we ran. We would rent out an entire bar. We'd have people come in and try these devices, and we try to understand their social acceptance and social perception of the devices and how they may use them in their natural world. So people will laugh. Some people will say, I like it, it helps me relax, but I would never wear this because it makes me look like I have this, you're stigmatized, right? And so we worked a lot on modulating arousal, looking for mechanisms of controlling the central governor, if you're familiar with that. But I was trained as a psychologist and working in engineering. I always went back to this one kind of law in psychology that it's kind of intuitive, and many of you have seen it, and it's known as the Yerkes-Dodson law, right? And so this is where your ability to perform is a function. I don't know if, I guess the screen's kind of a photon trap. I'm losing my laser beam. On the y-axis, you see that you have physical and mental performance with peak or high functioning at the top, and on the x-axis, you have levels of arousal from low sedated state to panic or anxiety. Depending on the task that you're doing, right? So many of you have seen the traditional Yerkes-Dodson curve. It's an inverted U curve. It depends on the task. There may be some tasks that you're optimized for high levels of arousal, such as hitting a baseball, versus falling asleep where you need to be less aroused to be able to function, right? So from an academic perspective or scientific perspective, this is with the company, we started studying and trying to determine whether we could control physiological arousal. We know that the trigeminal networks feed into some of the same networks that the vagus nerve will communicate with, such as trigeminal nuclei, nucleus of the solitary tract, but most interestingly, the locus coeruleus, right? And so this is your fight or flight fear response. The locus coeruleus fires in different ways. When you're highly stressed, it will fire in a tonic high frequency way. That's kind of like if you've ever lost your keys and you're running around the room and you can't find your keys and you can't think about where you left your keys, your locus coeruleus is firing in a high frequency tonic state. You're not gonna find a solution, right? To find a solution, you needed to fire with stimuli in your environment in a phasic manner, right? And then there could be a low frequency tonic manner that it fires in a different state, right? So what we showed in this paper is that when we have this trigeminal nerve stimulation device, we shocked people. So we electrically shocked them. So we wanted to induce fear in people. So they were in a shopping mall. Someone would approach them and say, hi, would you like to be part of a neuromodulation experiment? And they would say yes, like we'll pay you $25. They'd come upstairs, and then we would show them a video of nature, and they would see like a whale swimming, they'd see a tree, and they'd see a bolt of lightning. And every time they got to see the bolt of lightning, we'd give them an electrical shock. And so that was the fear conditioning component. And then we had another component where we would just show them the bolt of lightning without shocking them, right? So we trained them to this bolt of lightning. What we found is that there was a reduction in galvanic skin conductance that was timed to the electrical shock. There was an increase in facial temperature. So we use FLIR cameras or microbolometers to detect temperature changes. This is direct report of sympathetic activation, right? So you have a sympathetic nerve fiber that innervates the vasculature. When there's a decrease in sympathetic nerve activity, the blood vessel dilates and you see there's an increase in temperature, okay? So if you have vasoconstriction, the skin cools. That's why when you're stressed sometimes, your fingertips will feel like they're very cold, or the tip of your nose feels like it's cold. The tip of the nose is you can't lie, right? We can sit here and watch you with a FLIR camera, and if someone gets stressed, your nose will get cold, right? And it's usually you can't see it, but I mean, with FLIR you can. Okay. So then it became the question, okay, so what is the functional significance? If we want to enhance performance and we want to improve brain health, what is the functional significance of being able to tune the brain's central arousal circuitry? We started to make these observations. Again, we had people just coming in the lab, probably 30 people a day, and every 3 or 4 people we'd see this phenomenon where they would just fall asleep in the middle of an experiment. Okay, so they come in the middle of the day, and you can see this individual starts to nod off. And then here's another example. And they just start to nod off, right? And maybe they're tired, maybe they're sleeping. Probably we said, well, maybe there's something more to this. So we ran a study. This is it's about 100 people. I think it's 95 people. We only ever published it in bioRxiv. A lot of my work I only ever put in bioRxiv because it's like you've got to move from one goal to the next goal, and you don't have time to go back and fight peer review, and it doesn't really matter in terms of moving the translation forward. What we found is that there was a decrease in anxiety when people use the device 15 minutes before they go to sleep. There was a decrease in the number of wake-ups, a decrease in the WASO, wake after sleep onset time, decrease in the time awake. So 10 LF, this is low frequency. We worked with a lot of frequencies. What we showed here is that a low frequency shifted in the hundreds of hertz compared to kilohertz had a better effect on sleep than the high frequency stimulation. The other thing is that, again, comparing low frequency to high frequency, we found that people would wake up with higher cortisol levels. That's actually a good thing. That's a sign of restorative sleep, right? You reset your cortisol levels. If you wake up and your cortisol is blunted, you're adrenally fatigued, right? And so then we also saw there was a reduction in morning salivary alpha amylase. So then we had this picture. Well, it looks like we might be able to trigger sleep onset. Is there a way that we can improve even more beyond that? Is there something we're missing when we stimulate these sleep arousal circuits related to the trigeminal nerve? So we're modulating activity of the locus coeruleus and other brainstem nuclei that make part of what's called the reticular activating system. Basically integrates all sensory stimuli coming into your brain and your brainstem and then sends that on to the cortex for processing. But those are the sensory cues that keep you awake, right? The waking tone. Sleep is this is called the flip-flop model of sleep, right? So it's a bimodal, relatively unstable state. You're either awake or you're asleep. There's no in between, right? So from a physiological perspective, you go from wake to sleep instantly. And so what are the triggers that allow you to fall asleep? When we were digging through the literature, I found this paper, and this paper kind of changed our entire approach and view on vagus nerve modulation and trigeminal modulation. In the 1970s, they started to look at awake behaving cat preparations and stimulating the vago-aortic arch. And in this particular example, what you see, the top two graphs, so the top graph is visual ECOG, then there's electro-opticalgram, so basically monitoring eye activity, and then there's EMG signal, and then this is vago-aortic stimulation. The cat is awake, you see an EMG signal, and then as soon as they start stimulating, the cat immediately reflexively falls asleep. It enters a state of REM sleep, okay? On this side, the histograms show that there's basically a decrease in sleep latency. So you can see that the sleep latency is 1 to 2 minutes on average compared to many minutes, up to 7 minutes, right? So these cats would immediately fall asleep reflexively. This has been replicated many times now by other papers. One of the conclusions, one of the functional hypotheses, and I think we forget about this, we talk about enhancing sleep or, like how you improve sleep. We often forget this, is that you first have to enter a state where the brain is turned off before you can fall asleep. And so what is that state? We think vagal stimulation could be one of the key ways to trigger sleep onset. It's used in veterinary practice, right? So you can do these vagal maneuvers. They have cats that will show up at the veterinary and they have high heart rate and the cats are all stressed and anxious. and they do vagal maneuvers. It's an ocular cardiac reflex. You can massage the eye, and there'll be an immediate drop in heart rate. You can massage the nose. It essentially induces a mammalian diving reflex. So part of the mammalian diving reflex is when our eyes are submerged underwater, the pressure will cause bradycardia, or a reduction in heart rate, slows the heart rate to conserve oxygen. So it's conserved, these mechanisms are conserved across mammals. This brings us to vagus nerve stimulation. So this is how we got to vagus nerve stimulation. Before that, I wasn't even really aware that vagus nerve stimulation existed, right? Like I'd seen a couple papers about it here and there. So we started to explore vagus nerve stimulation, and this crowd is obviously familiar with vagus nerve stimulation, both implanted and non-invasive vagus nerve stimulation. There's two forms of non-invasive. One is transcutaneous auricular vagus nerve stimulation, and the other non-invasive form is transcutaneous cervical vagus nerve stimulation. Our goal at the time, so as we were doing those experiments, at the time I was funded by the DoD and had been working with some other agencies, and one of the other agencies, we spent quite a bit of time with them. This was about 2012, and they talked to us and they said, we'd like to have something that can help enhance foreign language learning. But if our individuals use the device, they can't know it's on. They can't know they're wearing something. And they literally mean like you cannot tell that you have a device on your body or it gets in the way, right? And so we then started to ask the question, well, how can we focus on making vagus nerve stimulation more useful? There was evidence in the literature that you could modulate these ascending brainstem networks, including the nucleus of the solitary tract, parabrachia areas, as well as the locus coeruleus, from a couple different groups. This was the first paper, and then some others after that. The major problem, however, and I don't know how many of you ever had auricular vagus nerve stimulation, is that the methods are not electromechanically designed to be optimal for human use, right? And so I don't even know what paper this is from, but this person, if a postdoc or grad student, should be thrown out of school immediately. They're probably zinc-coated screws from Home Depot, something you definitely don't want to pass current through and putting against your skin. But it's uncomfortable, right? So what happens, I was talking to Sandy and Jane earlier, and I was like, imagine if you're a language analyst, you're working for the government, and we meet you on campus of wherever university we're at. Hi, I'd like to bring you in to run an experiment on you today. We're going to try to enhance your Mandarin foreign language learning. We're just going to put these little earbuds or clips to your ears and modulate your brain activity. And then we want you to try it first, and the first thing you feel is an electrical shock or a biting sensation because of the mismatch between this hard electron carrier and the soft tissue of your skin. All bets are off, right? And so when people will use these, you may see a decrease in heart rate, but they will describe it as a biting sensation or it's not very comfortable. That's not very adoptable. It's not very scalable. And it certainly was not going to work in applications to help people learn sensitive task. So the first thing we did is, instead of targeting these different regions of the tragus, the concha cavum, different anatomical parts of the external ear, we thought, well, what if we just go in the external acoustic meatus and have electrodes placed in the ear just like earbud electrodes? And the answer to that is there is innervation by 3 cranial nerves: the facial nerve, the auricular branch of the vagus nerve, and the auriculotemporal nerve, or the trigeminal branch. So the area of the ear is innervated by the nerves. Then it became, how do we design something that has a polymer, a conductive polymer, to interface with the skin to make that interface more comfortable versus having a hard metal against the skin? And so this is a patent that in 2014, I wrote the patent, and then I acquired the patent from the company that I had worked with at the time a little bit later, and then we've continued prosecuting it since then. In 2016, 2017, I guess it was 2016, I got a call, and someone said, DARPA TNT, look at it. And DARPA TNT was a program that was announced by DARPA largely due to Mike Kilgard's work, which you should all be familiar with. The idea was this type of approach, which Dr. Welle will be discussing, which is a task-locked VNS effect. So it's really a time-dependent VNS effect. You're delivering vagus nerve stimulation during some task or some very temporally precise window to try to induce plasticity. We work on the top part and still work on that top part in terms of autonomic arousal, cognitive gain, attention, mood effects, and inflammatory responses. This is a case where you can use vagus nerve stimulation and just turn it on. You don't have to worry, other than going to sleep. If you want to go to sleep, you should use it before you go to sleep. But if you want to have a general reduction in arousal, you can use it any time during the day. So we were asked, I was part of two TNT teams, one with, at the time it was known as the Center for Advanced Language Studies, now ARLIS at the University of Maryland, to work on vagus nerve stimulation, designing and teaching them the methods that would then go on to be used, in some cases, to enhance foreign language learning. And then I had another a team that we partnered with USARIUM to enhance marksmanship, and then we partnered with Air Force Research Laboratories to enhance cognitive skills for remote pilots. We taught the methods. Again, we published in bioRxiv and just left it. I think it's 54 figures of EEG that you can comb through if you want. I couldn't walk away with any clear conviction that it was doing anything at one frequency versus another, or was doing this to heart rate or not. I just knew that, I've talked to Sandy and others about this, and the populations that we work with, we just get a, do you feel good or do you not feel good? This is a go, this is a no-go, and that's basically it. So a lot of the EEG and heart rate, it's hard to really understand what it does, but so the most important thing was the disclosure of the method, right? I then, in 2019, left the TNT teams. A new program manager came in. Our teams were cut, and we designed the device that's shown here for a large consumer electronics company. I can't tell you who it is, but you can figure it out. We ran a fairly sized trial, a double-blinded, placebo-controlled study using EEG polysomnography looking at insomnia. The trial worked out. We were supposed to be acquired, but COVID hit, shut down the health division, the company moved on. They then took the device and combined it with their headphones and a notch filter, and were able to treat tinnitus. And one of the reasons that is interesting is because the vagus and other nerves in the cervical plexus, C2 to C4, they project afferents to the dorsal cochlear nucleus, right? So you can start to bias ascending auditory information very early in the sensory stream. After that, after COVID kind of shuttered that health division, there was a gap in the field in terms of being able to have these devices that use hydrogels in this manner. And so we were contacted. I got another call and said, you need to look at this program called SEMI-NBMC. It's Nano Biomanufacturing Consortium. And we were awarded an OTA from AFRL to develop and design an over-the-counter general wellness auricular vagus nerve stimulation device because the customer was on flight status. So the customers were on flight status, they were pilots. You cannot fly and take a drug, right? And so this was a very distinct class of device they asked us to build. They went to another company, Electrocore, and asked Electrocore to make the device. They said no. We said yes. They said we want it to be black and have a skull on it. That is our logo. You can now look up the TAC-STIM device that came after this and look at their logo and compare it to ours. And I'll let you draw your own conclusions. But we were told we needed to be hands-free. It's called Brain Bolstering Resilience, Adaptability, and Intelligence with Neurotechnology, mostly to help enhance and manage stress during operational activities, okay? Brain buds compared to other devices in the field. So this device, Nuvana, this device was one that's used by some of the partners to enhance language studies. One paper showed that you enhanced language using the device. Another paper showed you didn't. One paper used hydrogel to contact the skin. The other did not. We originally evaluated the device and looked at the device and spoke with analysts about using the device. The way it's designed is it's a conductive rubber that you spray with saline, and you put a conductive rubber saline electrode in your ear. That wet sensation is one of these. That is, I'm not here to learn Mandarin today. I don't like the sensation. Okay, so other advantages, we do high frequency stimulation. A lot of this is to get around the sensation, again, because you can't know that it's on, right? If you know that it's on, you've now reached a level of arousal that we don't really understand what's happening and not just decreasing sympathetic tone. As Dr. Welle will talk about, there's a lot going on in the brain when you modulate these circuits. and we don't really understand it yet. So we're just trying to stay with just the reduction in anxiety or stress performance stress. We have worked with, we delivered our devices to Air Force Special Operations Command and have worked with Air Force Special Operations Command, and more recently have some customers in the U.S. Army Special Forces. We're told, and we believe this, that the device is a cognitive multi-tool. It can be used for multiple applications, multiple times. The way we look at it, there's lots of variables. So this goes back to the Yerkes-Dodson curve. You can affect the shape of the pulse, the width of the pulse, the frequency of the pulse, the timing of the pulse. All those variables matter, okay? When we tune brain buds at 1800 hertz, we know that the below sensory threshold, 2 to 3 milliamps, depending on who you are and other conditions, how old the earbuds are, it acts like a digital clonidine, right? Because one of the paradoxes is if you're stimulating the vagus nerve and that's activating the locus coeruleus, how do you get a reduction in activity? Why wouldn't you be aroused? Clonidine is an alpha-2 adrenergic receptor agonist. It's the opposite of a beta blocker, but has the same type of function, okay? So at higher intensities, if you're talking about alpha receptors, it would act like yohimbine, and if you're talking about beta receptors, it would act like albuterol. I don't know how many of you have had albuterol before, but the jittery sensation you get, if you get a high enough stimulus in the ear, you will feel that same type of sense. It may not be the exact jitters in terms of the phasic responses, but you're not going to like it. So shifting to clinical applications, we've recently shifted to clinical applications and are focused on 3 different areas, insomnia, depression, and chronic inflammation. It's called the deadly triple threat. I don't think I have to belabor this on anyone in the room. We're very excited about being able to collaborate with the Center for Brain Health in these areas because we've not had a way to measure or quantify brain health. We feel like we're impacting brain health, but we've not had a way to quantify or measure it and show people. Massive cost to the country. I think a lot of it is driven by chronic inflammation. It's going to take several studies to show, but I think if we can treat the inflammation with vagus nerve stimulation, some of these other things may be corrected. Recent studies, so these are unpublished studies. This one's undergoing review. We recently completed a study. This is an open label study that we conducted on women with breast cancer, stage 2, 3 breast cancer, who also have insomnia. Using the device just before bed after 2 weeks. I don't know what happened to that slide, but my PNG didn't turn out so well. By the Insomnia Severity Index and the Pittsburgh Sleep Quality Index, they both had sleep improvements that were equivalent to minimally clinical important differences. There's also a reduction in fatigue, because I can see that when I can't see the other ones. Like, take off my glasses. Oh, so this is sleep latency and sleep efficiency monitored by Fitbit. So all the women had Fitbit devices. I realize that's not as good as EEG polysomnography, but look, when you run these studies, having someone have to wear a polysomnograph device or an EEG device at home, they're just not going to participate in the study. You're better off saying, look, just wear a Fitbit and please use the brain buds. Depression also decreased, although these patients did not have clinical levels of depression. There was an improvement in depression scores. Fatigue was, I think, in terms of life quality, was the number one thing that we hadn't realized was a big problem for people with breast cancer, especially if you're on chemotherapy. You have no energy. And so that was the one life outcome that we felt like was really big. Before this, we had done some work when I was still in Phoenix on children, high-functioning children with autism. I had mentioned before that the nerves on the back of the neck are very similar to the nerves on the face. At the time, the design of the electrodes had a steel post, and we wrapped it with the hydrogel. Now it's a little bit different. That's conductively insulated now. And the children didn't like when the steel post would hit their ear. No one liked it. It's not just because you have autism. No one would like that sensation. They didn't like the sensation, but they tolerated the back of the neck stimulation. And so we did that and found that this is feasibility, but we found that there was improvement in anxiety, improvement in sleep problems, and some cognitive improvements. We're now following this up at UAB. We're going to be doing more and more studies on autism. We'll be working with the Center for Brain Health on that. We've combined vagus nerve stimulation with PT, physical therapy, for dysautonomia and Parkinson's disease. On the left-hand side, compared to sham, there was a decrease in systolic and diastolic blood pressure and heart rate. So there's a reduction in systolic blood pressure for sham, but it didn't occur for diastolic blood pressure. And then heart rate and diastolic were both decreased after VNS. So this is prior to exercise. They do this for 15 minutes. A lot of patients who show up with Parkinson's disease, they have dysautonomia, so they don't have the kind of the cardiovascular dynamics that they need to benefit from exercise. And so we're addressing the autonomic instability of the disorder more so than the movement scores. But when you paired VNS with vagus nerve stimulation, there was a long-lasting improvement in motor outcomes, right? So that lasted beyond just exercise alone, because obviously exercise is also good. So summary conclusions, autonomic nervous system modulation through the external acoustic meatus is possible using using vagus nerve stimulation through the external acoustic meatus. We can improve sleep and mental health outcomes, right? Whether that's due to improving sleep due to decreasing arousal, or whether depression gets better because you make people sleep better. We don't know. I don't know that question can really be answered. Either way, people get better. Parkinson's disease applications. This is not one of our largest. We're more interested in dysautonomia because dysautonomia is common across so many different neurological conditions, but we are using that as a model in Parkinsonian patients. Current studies, we're focused on inflammation in sleep and mental health disorders, as well as autism, and look forward to continuing our collaborative relationships with the Center for Brain Health. And funding has been provided by SEMI, NBMC, AFRL, DARPA, IST, my company. There's a lot of people to thank. My graduate students, Alex Vancho, Melissa Du, Alex Sartiev, and Christian Lopez Blanco have done the work on breast cancer and Parkinson's. And then along the way, there's probably 5 or 6 people that I can't name that have been very, very helpful to getting me to this spot and have been advocates and helped along the way. And so I definitely thank them too. All right, thank you.
All right. Really, really exciting work, Jamie. I've heard about it, we've talked about it, but it's always great to see it all put together. I also want to thank Sandy so much and the whole team for the invitation to come here. This is a really beautiful space with some clearly remarkable work going on here. So it's just been my pleasure to get to see everything that you guys are working on. So my talk's going to be a little bit different. My lab is primarily an animal model lab, and so we're very interested in mechanisms of vagus nerve stimulation that we can explore through the use of animal models, but we are launching some early clinical studies, and so I will, at the end of the talk, show some initial clinical findings, but kind of on a smaller and earlier scale than what we've just seen from Jamie's work. But my goal has really been to understand the mechanisms for vagus nerve stimulation and motor rehabilitation, and also we've made some interesting observations in how vagus nerve stimulation may influence myelin. And I'll tell you a little bit more about what myelin is and why myelin plasticity is actually so critical for long-term functional outcomes. So, I'm going to start just by introducing a device. This work is from a company, Microtransponder, which has the first FDA-approved vagus nerve stimulation technology for paired rehabilitation for stroke recovery. This work came out of work done right here at UT Dallas and the Kilgard and Renneker groups, where they did a large number of rat model studies showing that pairing vagus nerve stimulation with physical rehabilitation after stroke can improve motor outcomes. And so then that preclinical work was translated, and this company was spun out, Microtransponder, and they did the first clinical human study of this and found that, in fact, in humans as well as what had been shown previously in animals, people with stroke who, let's see, yeah, here we go. People with stroke who received paired vagus stimulation had better functional outcomes than those who just received physical rehabilitation alone. And what's important about this methodology in comparison to other uses of vagus nerve stimulation, so vagus nerve stimulation, for instance, is used to treat epilepsy and reduce seizure frequency. But in those patients, the device is implanted, a very similar device you can see here. There's a cuff that's implanted on the vagus nerve in the cervical region of the neck. But in patients with epilepsy, the device is turned on and it stimulates the nerve all the time. It's a continuous stimulation. And what's unique about this approach is that the vagus nerve stimulator is only turned on for really brief moments, and it's the moments when the person with a stroke is practicing those movements that are really important to them. And so the diagram here shows someone reaching for a water bottle and grabbing to give themselves a drink. And so the vagus nerve stimulation would just be briefly turned on during that movement. And that very brief application of vagus nerve stimulation leads to these results. And so my lab is really interested in understanding why this could happen. What are the mechanisms? There is some of the preclinical work done here has demonstrated that neuromodulators are involved, but we wanted to understand in a little bit more granularity what was going on. So we moved to our animal model, which is the mouse, and we teach mice to reach for a small pellet that's placed on a post. I will play that again. So you can see the animal reaching out, they reach for this pellet and pull it back in to eat the food pellet. This is actually a hard task for a mouse. It's easy to knock that pellet off, and so it takes a lot of learning. It's a dexterous motor behavior. And we wanted to understand how paired VNS might allow animals, these are healthy animals, to learn this task better. We explored a few different types of pairing to see which one was most effective. And so our types of pairing were what we call a random VNS. And this is just vagus nerve stimulation that's applied during this training session, but not paired with the specific reach itself. We also tried what we call reach VNS, and that is we stimulate the vagus nerve right at the beginning of a reach. And this would be reflective of spike timing dependent plasticity, so things that fire together, wire together. And then finally, we paired vagus nerve stimulation with success. So only when the animal actually grabbed that pellet and brought it back to its mouth, that's when they would receive the stimulation. And what we found is that there's only one condition that actually improved learning, and that was the pairing with success condition, which is shown here in orange. And so you can see on the left, this is a training profile over 14 training sessions. And you can see the animal's success percent. What percentage of their reaches did they actually get that pellet and bring it back to their mouth? And so in control is gray. You can see they have a learning curve, and then VNS enhances that learning curve above control. Interestingly, our pseudo-random stimulation, the one that's not paired with VNS but just applied during the behavioral training session, actually made the animals worse, as you can see in that light blue bar there. So we do the model that we have been working on since this set of experiments is that VNS is most effective when used as a reinforcer to reinforce the movements that were successful for that animal's behavior. So we continue to explore, what might be underlying this reinforcement mechanism. And we first wanted to understand what's happening in the motor cortex. So the motor cortex is a region of the brain that is responsible for these types of dexterous reach tasks. And so we used animals that are expressing a calcium indicator in their neurons that lets us track when each neuron has an action potential. And we can image this by actually removing a piece of the skull, putting a piece of glass in its place, and then using microscopy to look at those signals in real time. So we can kind of see these little pops of neural activity as the animal's learning this reach. And we have a lot of detailed analysis on this. I will cut to sort of the main point, which is that when we pair vagus nerve stimulation with success, our reinforcement VNS context, we see that the activity in motor cortex that reflects the outcome of the reach, so whether the reach was a success or a failure, is modulated by vagus nerve stimulation. And really only those neurons that care about success versus failure are modulated. All of the other neurons in motor cortex are not changed. So we think that there's evidence for circuit-specific modulation with VNS that changes the way outcome is represented in the brain, presumably allowing animals to access this outcome-related activity more quickly, more easily, and to learn better. We then also wanted to know how does a signal from the peripheral nervous system actually translate into the brain itself? And so we identified the cholinergic basal forebrain on the basis of prior literature as a likely key conduit that could translate these signals coming from the vagus nerve up to motor cortex. And so to test this, we did our same exact training protocol that I just told you about, but we had one change. We used optogenetics, which is a tool where you can modulate the activity of neurons with light, to silence cholinergic basal forebrain neurons bilaterally, but only for the same 500 milliseconds that we stimulate the vagus nerve. So the cholinergic system is working normally all of the time except for the 500 milliseconds of vagus nerve stimulation that occur when the animal has a successful reach. So just at the reinforcement epoch. And what we find is here, again, on these curves, gray is our control group, orange is our vagus nerve only group, and blue is vagus nerve plus cholinergic inactivation, shutting down those cholinergic neurons. And so, as you can see, the animals can't learn any better if we shut down those cholinergic neurons, suggesting that they're required. These cholinergic systems are required for the effects of vagus nerve stimulation on learning. So it was really helpful for us to understand that this is a cholinergic dependent activity. And so to sort of summarize this part of the talk, we've found that stimulation of the vagus nerve paired with success as a reinforcer can actually increase motor learning. It does so by elevating cholinergic basal forebrain activity, just very briefly, and it shapes the outcome response in primary motor cortex, which we believe is what's responsible for allowing this improvement of behavior and learning. There's more I could say on this topic, but I kind of want to just switch focus to another aspect of plasticity in the brain. You know, clearly there's neuroplasticity is involved in this accelerated learning, and we're continuing to study how cholinergic signaling might shape neuroplasticity. But there's another key aspect of plasticity that's often overlooked, but is growing increasingly important and recognized, which is myelin plasticity. Now, myelin is the substance that ensheaths the axons of neurons. And so it provides, if you think of an axon of a neuron as like a wire that transmits an electrical signal, this provides the insulation. Myelin is created by cells called oligodendrocytes. They're glial cells in the brain. And they actually stretch out their arms and wrap them around nearby axons, and that's what creates myelin. In motor cortex, axons are not continuously myelinated, so it's unlike maybe insulation on a wire. There's not just a single sheath. Instead, it's little patches. And these little patches, actually, it's a little mysterious why myelination is not complete, but one thing we do know is that by changing around where myelin is placed and how much myelin any one axon has, you can change the properties, the signaling properties, the communication properties of that individual neuron. So it's a mechanism to help to speed up or slow down neurotransmission or change timing in circuits. And one thing that has been shown by a number of other groups is that changing around this myelin pattern is critical for motor learning. Animals, if you block the ability to make new myelin and place it on sheets, animals cannot learn this task, the one I just showed you. They can't learn it. And so this ability to kind of change around myelination is really important, but sort of still being explored in terms of understanding exactly why it's so critical. But here's some more data from some other researchers showing that, in fact, myelin is changed dramatically over the course of learning. So we wanted to understand a little bit about if vagus nerve stimulation, we know it has an effect on neuroplasticity, but is it possible it could have an effect on myelin plasticity? And so we did this by sort of thinking a little bit about the process of myelination in the adult animal. One thing that's really interesting about oligodendrocytes, which are shown here in green, is that not only do they myelinate axons of local neurons, but they have a population of stem cells that tiles the adult brain. So they can be replaced. If one dies, a stem cell will differentiate and become a mature myelinating oligodendrocyte to take its place. Now, this process doesn't work perfectly. You may have heard of a disease called multiple sclerosis. This is a disease where oligodendrocytes in the brain die, and there are places, regions of the brain that lose all of their myelin. There is a recovery process in humans too. There are stem cells that create new oligodendrocytes, but for reasons that are not well understood, it's incomplete. So many people who have MS may experience a loss of motor function, cognitive function, sensory ability during a relapse when they lose myelin, and they may or may not recover that ability over the course of this remyelination process. So what we wanted to understand is, can we improve remyelination to help people with MS actually recover better from a demyelinating injury? We do this again with an imaging modality, so we can actually, again, take out a piece of the mouse's skull. We have fluorescent, oligodendrocytes are labeled with fluorescent indicators, and we can watch these oligodendrocytes die when we give the animals a drug that kills oligodendrocytes and then regenerate. We can watch new ones regenerate and watch what % of recovery is there and where does the new myelin go. And so what we do is we give the animals this drug called cuprizone for 3 weeks. This kills some of the oligodendrocytes. And then right after we take the drug away, they begin this recovery process. And that's when we perform some interventions. One intervention is just having the animal do motor learning, which is akin to physical rehab for people with MS. And we find that that does improve remyelination a little bit. The second intervention is to pair vagus nerve stimulation with that, just like we did in our healthy animal studies. So here's the, here is the data, here you're looking at the % recovery of oligodendrocytes over time, where 0 is when we remove the drug and the recovery process starts. And so in gray is our control. In green is our physical rehabilitation. And you can see it does improve that recovery of oligodendrocytes a little bit. When we add paired VNS, it actually improves it quite a bit more. So we get quite a bit more recovery of oligodendrocytes, quantified here. Perhaps even more surprisingly to us is we then looked at, we can actually track where every myelin sheath is going. So we get new oligodendrocytes, they go and they wrap local axons, and we can image the placement of each of those sheaths, and we can say, are they going back into new locations, locations that weren't myelinated before the injury, or are they going back into the same location that was previously myelinated? Are they restoring a myelin pattern? And what we find is that with our, which is now our control group, is our physical rehabilitation or motor learning group, about 25% of the sheets go back onto previously myelinated locations. 75% just go into new locations. But when we add paired VNS, we actually double that percentage. So now we have 50% of the sheets going back to previously myelinated locations. And we quantified this a different way, which I won't go through, but looking at this in a different mechanism, we find something similar, that paired VNS increases myelin pattern recovery. So then we said, okay, well, this is great, but what about function? How well do these animals actually recover their ability to perform the skilled reaching task? And so what we did here is we waited a long time to see, and then we checked the animals again about 10 weeks later, and that's what's in pink labeled as retrieval. So it's how well can they retrieve this motor memory that they learned right after demyelination. And in our animals that had motor learning alone, they could still perform this task, but over the 7 days of retrieval, they didn't get any better. They didn't show any evidence that these circuits were really well consolidated. But all of our paired VNS animals got much better over these 7 days, showing that we had created sort of a lasting motor memory, a motor circuit, and we hypothesized that this could have been due to myelin. And so we looked for a correlation between the changes in myelination and these behavioral recovery endpoints. And we looked at oligodendrocyte number and saw no correlation with success. We looked at sheath number and saw no correlation with success. But when we looked at our myelin pattern recovery, we see a strong correlation with success. So we really think that restoring the myelin pattern is actually important for long-term functional recovery. So it's not just about making new myelin. It's about putting myelin back in the right spots to enable this long-term formation of motor memory. Myelin pattern restoration predicts long-term motor memory. We tested this one other way with an animal that's genetically modified, so when we give them a drug, they can no longer make new myelin, and we got the results that we were hoping for, which is that in the animals that are wild type, we see the same sort of improvement at our long-term recovery metric, but the knockout animals don't. So suggests to us that we were, in fact, onto something, and that myelin does play this role. very quickly, I'll say that we also looked at what the neurons are doing when we apply VNS after a demyelinating injury, and this is actually looking at different types of neurons. And so when we look at our whole population of neurons and we look at their firing rate or mean ISI in blue, we don't see any differences between demyelinated animals and animals that demyelinated animals and demyelinated animals that are getting VNS. But when we actually look at break neurons down by whether they're excitatory or inhibitory, we see some big differences, such that the excitatory neuron firing rate goes way down, so we reduce hyperexcitability that's been shown after demyelination, and we increase our inhibition. So we think that VNS, even on the first day that you're applying it, it's shifting the excitatory-inhibitory balance in cortex and allowing for this improvement in recovery. So to summarize this point, we think that VNS paired with success can increase oligodendrogenesis, and it can modulate neural activity in a specific way, and then allowing sheaths to be targeted back to previously myelinated locations. Okay, so that was all the animal work. These are the insights that we're bringing forward, but what about, moving this into human patients? Let's talk about that. So Jamie did a great job explaining transcutaneous auricular VNS, but essentially there's a little branch of the vagus nerve that innervates the ear, and you can access it at various points in the ear. We choose to access it in sort of the concha region, which is like sort of this external part here where you might put an earbud. And we first wanted to know, okay, what happens in our mouse model? I'm sorry, we're not fully leaving mice behind yet. What happens in our mouse model if we do this in the demyelination context? And what we find is that, in fact, just stimulating non-invasively the concha of the ear increases oligodendrogenesis over control conditions. So we were able to show that maybe you don't need an implanted vagus nerve stimulator to increase your myelination, and the degree of increase was similar between implanted VNS, which is in blue, and transcutaneous auricular VNS, which was in orange. So this gave us some hope that maybe we could access these same mechanisms of at least oligodendrocyte proliferation using non-invasive stimulation. So we then went into the lab and started recruiting lab members primarily as our initial subjects, and looked at a few metrics just to say, can we in fact see any physiological differences when we stimulate here in the concha region of the ear? This was new to us, and we wanted to see, does this make sense? But we're in fact able to do a few things that replicate known findings, which is that we can change or decrease heart rate with these longer stimulation epochs, 60 seconds of taVNS. We could increase heart rate variability, which is another well-known effect of vagus innervation on autonomic function. And we could perform a slight modulation of the pupillary light response. So we see some evidence of influence on that. We then wanted to say, okay, what about cognitive function? Can we use a taVNS to actually influence how someone is performing on a cognitive task? And so we used a common task called a Stroop task, which is where there's a sort of a conflict between the color of a word and the meaning of a word. You all are probably pretty familiar with this. And I will say this data is very preliminary. It's not ready for publication. We would need more subjects to look at this, but these are our early findings. We do see that taVNS, which is here in the more purple color, increases pupil dilation during the response portion of the Stroop task. And it does seem to have some effect on response time, although again, I would say these are probably early and underpowered results. We then said, okay, what about our pairing paradigm? We really believe that reinforcement VNS is what's most important, but we needed something that was a test that was a little harder, so we used a Stroop squared task. I don't know if any of you have ever done this. It is actually really hard. You basically have a double transform. You do your Stroop once, and then again for your response, and it's a little bit of a mind meld. But in a very limited number of subjects, we, just a proof of concept study really, we paired taVNS whenever someone was able to answer correctly in this, which the correct rates are much lower for this test than a traditional Stroop. We see, maybe there is some evidence, although I would say we are very underpowered at this point in time to make these claims, but, maybe taVNS could help improve accuracy. And so we're continuing these studies in the lab. And then finally, and this is my last slide, moving back to a MS context, and we talk about myelination, we did, in fact, want to see if we can replicate some of the findings that we found in our animal models on fine motor and dexterous motor performance in patients who have MS and have impairments in upper extremity, or in fine motor function, actually. And in fact, about 75% of patients with MS do have fine motor impairments, and these impairments correlate directly with the quality of life for these subjects, as you might imagine. And so the design for this study is that we had people completing a grooved pegboard task, which is similar to a 9-hole peg test, but there's 25 holes and the pegs are oblong and so are the holes. So it requires quite a bit of dexterity to sort of rotate each peg and place it into the hole, and they vary in their orientation. And so we then had our participants complete this task, and when they were able to successfully place the pin, we had a sensorized board, and we would automatically trigger a taVNS stimulation. And again, super early results, but when we look at our just overall time to completion, we see that overall those who are receiving the taVNS were able to perform a little bit better on this task than those who are not receiving taVNS. Very much underpowered, and we are continuing these studies as well in the lab, so we hope to have some results that are appropriately powered soon. So with that, I'd like to thank the lab. We have some really fabulous people in the lab and our wonderful collaborators and funders. Thanks.
Thank you so much, Jamie and Cristin. That was absolutely fascinating, and I know we have a bunch of questions. Jamie, if you want to get up here, and we're going to take a few questions. I'm going to start with one that came in online. So, for the modulation of sleep and mood, was it actually all night or was it for a limited period of time?
No. So our treatment paradigm for treating insomnia and mood is to use the device 15 minutes before you go to bed, and that's 20 minutes before you go to bed. So you should use the device, go to bed, lights off. Right? What can happen and what could interfere is if people use the device 30 or 40 minutes before they go to bed, it's just sleep hygiene. Don't go check your email. Don't go turn on the TV and watch cable TV news, right? That's bad for your brain health.
Thank you for that answer. Do we have any questions here in the group? Let us get a microphone to you, just a sec, because we have people online, just a sec.
I'm just curious as to the application of this vagus nerve stimulation to maybe early onset dementia, particularly prefrontal anterolateral dementia, possibly it's delaying the progress. If you thought about as an application.
So that's something we've been discussing with Dr. Chapman and the Center for Brain Health, right? One of the things that we'd like to look at is more cognition, using taVNS to prevent cognitive decline. And so whether it's Alzheimer's, dementia, or some other cognitive dysfunction, but it's not something we've looked at personally yet.
Yeah, likewise, we have not looked at that, but we are going to be, after we complete our taVNS studies in healthy subjects, we are going to be working in Parkinson's subjects who have some cognitive impairment and trying to translate this set of studies into that population. But I think, just like Jamie said, this fits in so well with the ideas that we've all been discussing as a part of our collaboration with what's going on here.
To add on that, since you brought up cognitive impairment, that's something we run into at UAB in our studies with Parkinson's. We have patients oftentimes, they won't score high enough on a MOCA to be enrolled in the test because they won't understand the instructions. And so it is something you need to take into consideration. I do think that the Brain HQ and Brain Health Index will be very helpful there.
Jen?
So you guys have talked about, I love, my question's about optimization of function. So I love the rat studies. I love the response also in disease. What is the implication for people thinking about tip of the spear folks? How can what's the implication for this for being able to be faster, sharper, stronger?
So that is a loaded question. Because I think, and again, doing a lot of this work for the DoD for a long time, there's this idea that you're going to build super soldiers. Your superpowers are within yourself, right? You get in your own way. So we think about free throw shooting in basketball, for example, right? A professional NBA player should be able to sink a free throw every single time because the motor memory is there. It's the thought between your ears that gets in the way. And so that's what we're trying to deal with. We're not trying to make you faster per se or shoot better. We're trying to take away the self-talk and self-doubt that prevents you from performing at your best.
So I'm interested, Dr. Willie, and both of you, in the myelin changing, because we've looked so much at functional connectivity. And so tell us, I mean, as we're looking at optimization, the type of measures, what we should be focused on is people get better, optimizing it year by year. What do you think about these, kind of what the sophistication of your work and this vagus nerve stimulation to make our brains younger than they are so we don't lose myelin? I was really interested in the fact of replacing it versus just randomly to make sure that that's happening. So that was really curious.
Yeah, I think this is a really important point, and I think that generally the role of myelin in maintaining and supporting our neural circuits is underappreciated. I think it's a critical component for forming healthy, vibrant, long-term memories and circuit connections. And so what I would anticipate, what I would hypothesize, is that as we use training protocols that enhance cognition and maybe, optimize processing speed in the brain or memory, that the tracks, the white matter tracks and connections that underlie those functions, I imagine that they should increase. I think that we should see changes. Now, quantifying myelin in the brain is a challenge. There are, MRI-based metrics, but they tend to capture large-scale changes better than sort of smaller and more subtle changes. That's why the animal models are so nice in this. We can really get down to a very granular level. But I think that what we find, so I'll actually give you one more piece of information that I didn't present today, is that VNS alone can boost oligodendrogenesis on its own. It doesn't have to be paired with rehab. But pairing with rehab is the only way to get that myelin pattern replacement. And so that's why I'm a big proponent of using VNS, at least for this context. Now, it depends on your indication. If you're talking about sleep, applying VNS right before sleep makes a lot of sense. But if we're talking about learning, I think that pairing VNS with what you want to learn is really critical.
And so 2 things on that. So we're excited to collaborate with Dr. Welle at UAB because we'd like to start combining some of her methods in our Parkinson's studies. The variable that I think is really important, Sandy, that you mentioned is task difficulty. So that's something we've talked about. We talk about this a lot in terms of flow state. You'll never reach a flow state if you're doing something you're capable of doing. It has to be something that you cannot do to reach a flow state. I think it's probably the same with VNS. If you're just doing this like the Stroop task, right? Like if the Stroop task was not hard enough, you're probably not going to see some of the same type of plasticity that you may see otherwise.
All right. Well, thank you all so much again for sharing your truly groundbreaking. Oh, yeah, sure. It's your party.
From our perspective, I think it's accessible because a lot of times, when people think about brain function, they think, oh, I may have to go get an MRI, or I may have to go get EEG, I may go see a neurologist, right? So I think it's accessible the way that it's built for everyday consumers, right? That's why I think it's helpful, because if I give people the NIH toolbox and I'm like, this is your way of measuring your brain health, they're not going to be interested in the study. They're just not going to engage. And so I think the biggest thing that you've got is that it's accessible and people can understand it and relate to it easily.
Yeah, and I would build on that just by saying that what I really like about the Brain Health Index is that it's broad and comprehensive. And so, our goal is not to make any person better at the Stroop task, right? Like that's not a sort of generalizable skill in life. Instead, what we want is to be able to train on a set of training tasks so that we enhance like a holistic perspective of brain health. And the Brain Health Index is fantastic for capturing that truly holistic measurement.
the Brain Health Index for a while. But the other thing is that it breaks down, like, it looks at brain health as different components, so you get a holistic picture, right? Because, like, we think about brain health, sleep is always the number one thing I look at. It's like, look, are you sleeping well? If you're not sleeping well, you probably have poor brain health, you probably have depression, you're probably anxious, maybe some addiction. But I think if you can break it up into the components, because I might not be sleeping well, but if I have a better friend and support network, my emotional connections, social connections are more important that way. And I think that's what's, it's a composite, right? Like it's hard to explain, but it's just a composite of like, there's different things in your life that you can do to improve your brain health. It's not just one thing. And you get a score for each one of those.
All right. Well, I think that it's very apparent now that these lines of research highlight how mechanistic discovery, device innovation, and behavioral neurosciences can converge to really reshape our understanding of brain health, and that collaborations such as this move us closer to scalable strategies that can improve the brains of those who are facing disease and decline, as well as those who are navigating normal aging. And by working together, we aim for this collaboration to create a framework that can be implemented broadly and change clinical practice, making lifelong brain health a reality for all people everywhere. Because lifelong brain health, that is our shared mission. So let's give a round of applause for our wonderful speakers. And again, thank you all so much for joining us today, and we hope to see you again on October 10th when Dr. Patrick Stewart from the University of Arkansas will talk to us about sidewalk neuroscience to study power relationships. Thank you so much and have a wonderful day.